at 7 o'clock. But again, we're just gonna give those other folks a few minutes to join us. They were aiming for 7:05. Hopefully they're kind of coming in. So welcome everybody. Welcome to 2022 and Happy New Year. I know we all wish we were meeting in person, but we'll get there. I do think we'll get there. And we've gotten pretty good at this, and I think we can make it work. I do want to welcome the new members. Again, for whatever reason, I'm not seeing everything, but I'm hoping some of our new members are out there, and hang on, we'll get to meeting in person one of these days. If you saw the newsletter or were at the last meeting, you know that some folks have gone off the board, and I know I've said it before, but I just want to say one more time, since it's 2022, I do want to thank our outgoing board members. Cheryl Jacobson, what an amazing job. I hope Cheryl's on the call. Great job as president for 4 years. We had Tori Gussman and Dean Shostak going off the board for programs, and they got us through a lot of COVID programs. And then, of course, Bill Williams, who's gone off the board as past president. We're not going to let Bill get too very far away, though. We need his advice and his counsel. So I know all of you are still around, but thank you all so much. And Patty Maloney is our new vice president for programs. Patty, unfortunately, isn't able to be here tonight. She had a kind of a last minute family issue she had to deal with. So it is my pleasure to be able to introduce Dan, somebody who seemingly doesn't need any introduction, but I will say I was sent this little bio, and I learned a few things that I didn't know. So I'm going to go ahead and read this about Dan Cristol. Dan has been teaching biology at William & Mary for 25 years. He is an avid bird watcher and just raced to Maine to see the Steller sea eagle. Very cool. His research began as basic academic research on the evolution of bird behavior, such as optimal foraging, migration, and pecking orders, but has shifted over the years to more applied subjects such as the effects of golf course management techniques or mercury pollution on birds. Dan has published 100 scientific articles and mentored over 200 students, including dozens of graduate students supported by Williamsburg Bird Club grants. This is amazing. Locally, he has written 240 consecutive monthly columns on birding for the Virginia Gazette and covers the Jamestown area for our Christmas and spring counts and co-compiles the Cape Charles count. So all of that while he's been an incredible friend, advisor, and supporter of the Williamsburg Bird Club. And so we are thrilled to have you, Dan, as our first presenter for 2022. And I will turn it over to you.
Thank you very much, Nancy. It's a real honor to be here and hopefully fun as well for me as well as for you. I've tried to put together a program tonight that is entertaining and doesn't have too many graphs in it. And I know I achieved the not too many graphs part, but we'll see about the entertaining. I have been involved with the Bird Club since arrival 25 years ago, and the club was already quite mature at the time. It's been fantastic to grow and see the changes. I've probably attended almost every meeting for the last 25 years. It feels like I have anyway. And I certainly hope that we can get back to meeting on campus in person. I don't expect that to happen anytime soon, but I do want it to happen again, as we have for all these years. Tonight, I'm going to show you a little bit about my life and how I got interested in the things I'm interested in. Then I'm going to run through, for a few seconds each, all the studies I've done at William & Mary, but without graphs. Most of them, I'll just narrate them. And then I'll end up in more detail on the work that I've been doing that's shown here with mercury, which I've been has been the main focus of my work for the last 15 years. This is me standing in the South River, which is a tributary of the Shenandoah River. Behind me is a factory which pumped a lot of mercury into the river accidentally back in 1929, and the mercury's still there today, causing a lot of harm for birds. And so we'll get back to that later in the talk in some more detail. But here we go. I'm going to show you some pictures of me when I was young, because that's what people do. Let's see, click on that. There we go. Okay, so this is a picture of me in around 1969. I was already a bird watcher at the age of 4 or 5. And my spark bird, the first bird I saw ever, you may have to move your little box of pictures. It may be on top of this American woodcock picture here, but that is an American woodcock. That's not the one that actually I saw when I was 4 years old, but I had been suspended actually for inciting arson at my preschool, getting 2 other kids to start a fire. And so I was home for a week, and it was in the fall. We had a little tiny backyard with bricks, just like the one shown here. And I looked outside and I saw a bird just like this, sitting just like this on the bricks. And I called my father, who was a bird watcher, and described it. And he said, you know, there's a bird, looks like a football with a beak. And he knew what it was and told me, and that is the first bird that I ever got on my life list, I guess. I've been bird watching ever since. So it's not only my vocation, which I'm going to be talking about tonight, but it's also my avocation. I spend most of my free time reading about birds or going to look for them. So that's me when I was 5 or 6 years old. And there's me in high school with my best friend Nathaniel Kahn, who I've been friends with ever since, and that's who I just went to see the eagle with, the Steller sea eagle. There we are, clearing some habitat on an island in Maine for birds. Noticed the full heads of hair, and we were willing to take our shirts off in public back then. I won't say much more about that, but it's kind of amazing how bad photographs were in those days too. I just realized while putting this together, all of my photographs before the cell phone photographs were really terrible. That's me in college. I took ornithology. I went to Cornell, but I didn't take ornithology there. I took ornithology at Evergreen State College. I took a semester off and went out and took a field ecology course, a field ornithology course. And we just spent the entire time out in the Oregon desert, and it was an amazing experience. So that's me in college. Here's me in grad school. In the middle here, again, without the shirt on. I spend all my time with the dermatologist now as a result of all that shirtless activity as a kid. I worked for the Nature Conservancy for a couple of years, and then I went back to graduate school. I kept working for them for the first year of graduate school. And if anyone knows Tom Smith, by the way, who's the director of natural heritage in Virginia, this is Tom Smith. He actually worked in that office then. He's now a big shot in Virginia State Conservation. So my birding has taken me all over the world. There's a trip to Australia with a crimson rosella perched on my shoulder, helping me identify it. But primarily, I'm a local birder. I really enjoy the Christmas counts, and I go out walking for an hour every single morning on my street, and that is where I do most of my birding. And just seeing the slight changes from day to day, or the lack of change, seeing the same bird in the same place, that's really what I like. But every once in a while, something really unusual walks across the road. This is Bill Williams' picture of the limpkin this very limpkin is in my neighborhood. And so it's a lot of fun. Birds really, the potential for something like this can really make every day exciting, every minute exciting, really. I do chase rare birds. My photography is terrible. This is my photograph of a flamingo. You can barely see it there, that I went to see in Florida. That same flamingo is still there, by the way, if you need it on your life list. It hasn't moved in 4 years. And just this week, I went up to Maine. Again, not my photograph, as you can tell, because it's in focus. That is the Steller sea eagle that everyone's talking about, that is up in Maine currently. Came over from Siberia, was seen in Alaska, was seen in Texas, showed up in Quebec for a while, then Nova Scotia, then Massachusetts, and now it's in Maine. So maybe it'll show up in Virginia if we wait long enough, but I wouldn't count on it. I hope he makes it home. So I do a lot of birding. But tonight I'm going to talk about my research. Here's a little bit more about me. That's my family on the left. My two daughters, both named after birds. Indigo is next to me and Lajla is next to her. My wife, Rebecca. There we are in California. And on the right is just an example of what you could call my academic family. I used to sort of not believe in this thing when the older professors would talk about their students as if they were sort of somewhat like their offspring, but I sort of feel it now that I've been doing this for a while. This is just an example. Both of these students, Emily Snell-Rood on the right and Megan Cobiella on the left, they both received Williamsburg Bird Cub grants when they were here. Then Emily on the right is now a professor at the University of Minnesota. She's incredibly successful, far more successful than I am. And then Megan came along at William & Mary, and then she went and worked with Emily as her PhD student, and now she's a professor at Sweetbriar back here in Virginia. So you just see these, the fruits of your efforts at work, producing more researchers, more bird watchers, more good citizens. And after 25 years, I'm finally starting to sort of see what that's about. It does take a while, though. So when I first got to William & Mary, this is the study that I did. This is the research that I was doing. I had been in California studying crows and how they drop walnuts and break them open. And so it's avian prey dropping behavior. When I got here, there are no walnuts for the crows to drop. So I looked at gulls, herring gulls specifically, dropping clams. And I'm going to go into a little bit of detail on this study. This is one of in the introduction, Nancy said that I did basic research and that I've moved into applied research. So here's an example of the kind of basic research I was doing. When gulls pick up a clam, they can't eat it. It's too big to swallow. They can't pry it open. The clam's alive. They pulled it out of the mud. So they have to drop it on a hard surface, which around here is tough. They have to find a road or a special area to do that in. And if they drop it successfully, it will break open and they can eat it. Someone might steal it. It may not break open. They may have to do it over and over again. And so I studied this with the idea of the simple question, have birds evolved to drop their prey efficiently? In other words, is their behavior the product of evolution? And if so, what are the factors that they are optimizing or making efficient? Is it the height of the drop? Do they use the least amount of energy possible to get the most amount of calories? Do they pick exactly the right sized clam? How has evolution shaped this exquisite and seemingly what you might call intelligent sort of behavior? Or is it? So this next graph is terrible. I'm going to talk you through it though, so don't be upset. But first I'm going to ask you to read everything below it, and then I will talk you through everything that's written below that. This is a graph out of one of my papers that I wrote about gulls dropping clams. So take a minute and read that. All right, so I read through that, and I'm a slow reader, so I hope that you're finished too. Now I'm going to walk you through it. But basically, first I want to impress you with the fact that this work, which sounds kind of exciting, when you really get down to it, it's kind of dull. First you sit out there for, this project took 6 years, so thousands of hours. You're just sitting there in the cold, watching these birds, writing things down, videotaping them, and then you write these papers and make these calculations that are dry. So let me explain to you what's going on here, because it's actually very exciting. The question is, have the birds evolved to drop these clams efficiently? And so to do that, I had to figure out a few things. First of all, how much energy can they get from a clam? So this dashed line that says grams of meat, as along the bottom you can see size class, so that's how big the clam is, and the bigger the clam is, the more meat it has. That is not surprising at all. So that's the line that starts at 1 and goes up to the right. So the bigger the clam, the more meat it has. We had to, of course, collect hundreds of these clams and weigh them and dry them out and measure them, figure out how much water was in them and how much meat was in them. Okay, the next line is the all black one, ease of breakability. Now you might think that clams either are all equally breakable or maybe they get more and more difficult to break as they get bigger, but actually it's a complex relationship. When they're little, they don't weigh very much. And so if you drop them, which we did, we dropped hundreds of them off of Millington Hall to hit the bricks, the way the birds do, from different heights. When you drop them, if they're light, they don't hit the ground very hard and they don't break. If they're really heavy, they are stronger. And for this graph is, as it says there, it's 11 minus the drop height that it took to break the clam. So if you look at 6, there's this big dip at 6. 11 minus that means that for some reason, 6 is the perfect thickness for a clam not to break. Okay, the higher, it's not what you'd expect necessarily, but we dropped hundreds of each size of clam, and a 6 is thick enough, but not heavy enough perhaps, that it's hard to break. A 7 is much better. A 7 is heavier perhaps, and will shatter more regularly. An 8 is even better, but there's a problem. 8 is getting pretty large and hard for the gull to carry. So we did another measurement, which is I asked the bridge policeman at the Chesapeake Bay Bridge Tunnel to escort me across the bridge, and I picked up a bunch of those dead gulls that are always out on the bridge there that had been killed the night before, and I cut off their heads, and I put their heads into these shakers, and I put a clam in their beak, and I shook them like this, and I measured how long it took for the clam to come out, and I put 10 of every different size clam in their beak. And what that does is to just tell you which size clam is a naturally best fitting clam. And what you can see from the dotted line that drops is that once a clam gets to be size 8, which is nice and big, it doesn't fit very well in their beak, and it comes falling out. So this was an attempt to see if there was a live gull trying to transport a clam from the mudflat over to the place that it had to drop it, which would be some rock or some road somewhere. What's the likelihood of it falling out? So take a look at these 3 lines and figure out what is the best size clam for a gull to eat if it wants to break it easily and get a lot of meat. And you can't really figure it out mathematically from looking at the graph, but if I point out to you that the bigger they are, the more meat they have. Size 7 is much more favorable than size 6 in terms of breakability, so it breaks easier. And anything above a 7 is very hard to handle. Then you might conclude that the gulls would benefit most from eating size 7 clams. Easy to carry, easy to break, decent amount of meat. You can see that the amount of meat actually jumps a little bit between 6 and 7. So in this next graph, we see what they actually eat. I'll give you a minute to read this. And so based on those calculations, we predicted they should eat a size 7 more than anything else. So we sampled clams from the mudflat and saw what was available, and that's what's shown in the white bars. So you can see most of the clams are size 5. That's the most common size. That's the highest white bar. And then we picked up clams that were broken on the road at Jamestown Island, thousands of clams. And we looked at their sizes. And what you can see is that a size 7, the difference between the white and black is biggest on the size 7. In other words, they eat a lot of size 7s, even though there aren't that many out there. So they're out there seeking size 7s, and we were able to predict that that's what they should do. So this is just a taste of what my research was back in those days. And also, I wanted you to realize how complicated and sort of boring these graphs are, so that you didn't mind that I didn't show you graphs for all the rest of the studies until the very end. And you'll be happy just to have me narrate them. But this gives you a taste of the kind of work that I was doing when I got to William & Mary 25 years ago. Another study that we did for 8 years with John Swaddle and lots of graduate students and undergraduates was asking the question whether golf courses are good places for birds. It's not an obvious answer. We think there's lots of birds on the golf courses. We put out birdhouses on the golf courses. They're often nice and bright green, and they're sometimes surrounded by cities and houses, so it seems like a good place for birds. But they are also really full of pesticides, herbicides, and fertilizer, and lots of human disturbance. So we looked at all different aspects of how well bluebirds, which nest readily on golf courses, do on golf courses versus other areas that look like golf courses but don't have the chemicals or the usage. And that would be college campuses, parks, places like Newport News Park off the golf course there. So we first looked at what kinds of birds are on golf courses, how many. We looked all over this part of the state, and we found that golf courses do have lots of birds. They're comparable to residential areas and agricultural areas. They're not as good as intact forest. Then we looked at how well the birds were doing, in particular the bluebirds. We counted their eggs, we looked at the dates of their laying, the size of their babies, how many babies they had, and we found that they were doing as well or better on golf courses than off of golf courses. So we didn't see strong effects of the pesticides or the fertilizers or the herbicides or the disturbance. We even looked for the pesticides themselves, and there's really not much evidence that they're getting exposed to pesticides. That's a tough one because the golf courses just use them periodically, and they won't necessarily tell you what they used or when they're going to use them, so it's hard to get out there right after they've sprayed. But golf courses got a pretty good bill of health, and we wrote a bunch of papers about that, and we also wrote a lot of articles in the golf course trade magazines about how they could design their courses to have more woodland. But I was curious as to whether maybe we were missing the whole effect, because maybe it was not the baby birds in the nests, but the baby birds after they left the nest that really suffered. And so we radio tracked, we put tags on the backs of baby birds, baby bluebirds, and we tracked them for the whole rest of their first year. This is Allison Jackson with a radio transmitter antenna. And we looked at mortality rates, which are extremely high for these birds. You know, if you look inside nests, you get a pretty good feeling about how birds do. They mostly survive to leave the nest. After that, it's all downhill. Most of them die within the first 3 months of any kind of bird. We thought maybe that would be higher on the golf courses. It actually wasn't. The birds on the golf courses did okay, but they did okay off the golf courses. They all left the golf courses right away and moved into the woods right around the golf courses, and we were able to track them there. So essentially, golf courses were much better than I expected, and it's always great when your results surprise you, because I really went into this thinking golf courses were bad, and I came out thinking if designed right, they're actually can be good places for birds, some better than others. So that was a line of work that was moving into the applied area, because we were trying to make recommendations for golf courses, and that was my first taste of really applied research. It's much more appealing to students, so I've been drawn more and more into the direction of applied research because that's what students are interested in. I've had about 200 undergraduates do research with me and 40 master's students. We don't have a PhD program in biology at William & Mary, so they're all master's students. And you know, the research I do generally is based on what students are interested in. So that was the golf course research. After that came a whole long series of other projects, which I'll just quickly work through here. I've talked about optimal prey dropping. I've talked about golf course design. Next set of studies we did was on the hippocampus and migration. This is basic research, which is all I used to do. That figure up in the left corner is a slice through a brain of a bird. And that part there, that's sort of like half a heart shape, that's the hippocampus, which is the part of the brain that is involved in remembering, in spatial mapping and remembering where you are. So work I did before I came to William & Mary showed that birds that store their food, like chickadees, titmice, they have much larger hippocampus because they have to remember thousands of places where they stored food, and so they've evolved a larger hippocampus. I was interested in whether birds that migrate and have to remember different places that they stopped, whether they also have a larger hippocampus. So I worked with these juncos, and Virginia is the perfect place to do this because in the mountains of Virginia, we have non-migratory juncos that It's a different subspecies. They have been separated since glaciation. They live in the high elevations of our mountains, and one could compare their spatial memory, their ability to remember things, their hippocampus, to those of migrating juncos, which spend the winter with them. And you can catch them together in the winter. And so what we found was that the hippocampus of the migrants is got more cells in it. It's not bigger, but it's got more cells in it. And they're much better at solving memory problems. When you bring them into the aviary, they can remember where things are better than the resident birds that don't migrate. And so this is a nice example of how quickly the brain and behavior can evolve, because we know that these two birds are only separated by about 10,000 years of evolution. And since the glaciation, since the glaciers retreated and the migratory birds started migrating up north, they have no longer bred with the resident birds. So that was an exciting line of work, and I haven't really finished it. We have one interesting study remaining in which we look at whether the actual act of migration causes the hippocampus to get bigger, and it does seem to. Bird hippocampus is unlike a lot of other parts of the brain. They stay changeable throughout life. They get bigger and smaller with the seasons, and it looks like they get bigger after you migrate. So maybe I'll finish that study someday. Another line of work I did was created wetlands assessment. When a developer expands their parking lot or builds a new store or something, and they destroy some wetland, the government requires them to build a new wetland somewhere else that's a little bit bigger, so that we don't lose our wetlands. This is mainly for flood control. We need wetlands for flood control and other functions. And so if you destroy a quarter acre somewhere and build another acre somewhere new, then hopefully the region is okay as far as flooding. But as far as birds, I wanted to know if those created wetlands actually provided replacement habitat for the wetlands that are lost. And so we looked at a series of created wetlands that were 5 years old, 10 years old, up to 35 years old at that time, and looked at the birds present in those throughout the region. And we found that the birds are not really returning to the wetlands in a comparable way as they had been present at the wetlands that were destroyed. So while it may replace some of the flood control services of wetlands, it's not replacing the bird habitat. And that work was also pretty applied. So I was beginning to do more applied work there. That was exciting work and probably could continue. There's a lot more wetlands have been created since I stopped doing that. Another study that I did for several years was on feather degrading bacteria. This was really interesting work, and it came out of just a conversation that I had. This is how a lot of these projects start. A student was interviewing with another professor and I, and he was a microbiologist, and we just started talking about a paper we had read about bacteria on feathers that degrade them, break them down. And how is it possible that birds are covered in bacteria that can break down their feathers, and yet they still have feathers? And so we started a series of studies on this. And it is true, if you take a feather and you put it in a test tube with these bacteria, which are called Bacillus licheniformis, that are found all over birds, the feather will disappear. If you keep it in a salty liquid solution under warm temperatures and incubate it, the bacteria will completely degrade the feather. Now, this is surprising because feathers are hard to degrade. Under dry, in a museum, feather will last forever. In fact, the chicken industry was really interested in this Bacillus licheniformis because they produce probably trillions of waste feathers every year, and these feathers don't break down easily, and so they have a real problem on their hands, and they wanted to know if they could maybe find a bacteria that could break them down. So we worked with birds in the aviary, and what we did was to put these feather degrading bacteria all over them and have them live in the aviary and see whether their feathers got holes in them or degraded at all. At the same time, we took feathers of the same kind of bird and put them on the wall of the aviary and coated those in feathers so they weren't on the live bird. So we wanted to see, is the bird doing something to keep these things from breaking down its feathers? And that is in fact what's happening. That there's several reasons why these bacteria do not break down bird feathers unless you put them into a test tube. One is that there's other bacteria and yeasts on the birds that kill these bacteria. The second one is that birds coat themselves in oil from their preen gland. The one skin gland they have is right at the base of their tail, and birds are always dipping into that and smearing that on their bodies. And that oil contains lots of antibiotics, essentially, mostly in the form of other bacteria that kill these bacteria. And finally, ultraviolet radiation kills these bacteria. And so when birds sun themselves, they kill the bacteria that would break down their feathers otherwise. So it is a real problem for birds to have these bacteria on them, but they have many coping mechanisms, and so they don't cause a problem. But they also molt their feathers once or twice a year, and that would get rid of a lot of these bacteria. So that was exciting work, which I don't do anymore. A mix of applied and basic research. We were really interested in the basic part, but there are some applications there for industry. Another topic that I've studied off and on over the years is differential migration. Differential bird migration is, the differential refers to when ages or sexes of birds migrate different distances from each other within the same species. So for example, if you go down to Florida right now in the winter and you look in a marsh, you'll see a lot of female red-winged blackbirds. You won't see very many males. That's because the females migrate farther than the males. So when you get to the southern end of the range, you only have females. And the northern edge of the range, you'll only have males in the winter. Now, of course, they all go to the same places to breed. So the question is, why do they migrate different distances? Are the dominant males forcing the other ones south, or are the males forced to stay up north so they can get back to the breeding grounds early and claim a territory? So that question hasn't been fully answered, but dark-eyed juncos, shown here, are a great species for that, because both males and females and young and adults all migrate different distances. So in any place, in any part of the continent, you're going to have a different ratio of them, and one can try to figure out what it is about their social hierarchy and their pecking order that determines their migration distance. That's a problem. That's a basic evolutionary problem, an ecology problem. Doesn't have much application. People have been working on it for 50 or 100 years, and they probably still will be for another 50 or 100 years. But it's an interesting problem that some students take to, and you can use banding data from the national lab, or you can create experiments where birds can move different distances. And with all the new technologies for tracking birds, I think a lot of progress will be made in differential bird migration. So I'm excited to see if people can crack that one. We looked at white-throated sparrows here and found that there is differential migration in white-throated sparrows. We're right in the middle of the range, so up north there'll be more males and down south there'll be more females. But they're very tricky because they have two different morphs, and they're hard to tell what sex they are out in the field. There's yellow, there's the black and white ones and the brown and tan ones. So finally, and I'll spend more detail on this, is my work that started in 2005 with mercury and bird health, all different aspects of that. Of the 100 papers that I've published at William & Mary, 50 of them are on mercury and birds. And so this really has occupied most of my time in the last 15 years. And so I'm going to go into a little bit more detail with two graphs about what we found. But obviously, 50 papers worth is a lot. And so I'm just going to tell you about some of the highlights. Let me give you a second here to catch your breath. All right, now here's just a picture of some students. Again, move your little bar of faces if you want to see the two pictures on the right. Look at on the left, look at the excitement and wonder in this student's eyes as she releases this tree swallow. You know, I just know that wherever Elena is today, she can remember this moment. I mean, it's really made a difference in her life to just get out in the field and do something completely different that she'd never experienced in her life, and spend the summer doing that. And look at the upper right, look at the concentration on those students' faces. They're just completely experiencing flow, I think, here as they get involved deeply in this research in the laboratory. And look at this guy, Ravi, on the bottom right. I mean, I don't know how to describe the look in his face, but I was with him that night. And the idea, if you told him earlier in the day that he was going to be catching his own owl and taking blood from it and putting a band on it and releasing it, he would never have believed you. So these are life-changing experiences that these students have, and that's really what we're doing here at William & Mary. I mean, I am trying to move forward our understanding of ecology and birds, but primarily I'm a teacher educating these students, and so all the research I do is with undergraduates and a few master's students. And the reason we do this is to educate them. And the Williamsburg Bird Club has been extremely generous over the years, funding 3 or 4 scholarships every year to help these students with their field work. So I want to say how grateful I am for that, and I hope that the other clubs do that in their area with whatever university they might be nearby, because it's a wonderful thing. Even the $600 that each of these students get makes a huge difference for them. It doesn't sound like that much money for a science project, but that's makes a big difference for covering the cost of gasoline for their car or parking permit for campus or whatever it is that they have to do for their research that they wouldn't be able to do. So just wanted to take a moment and show you some of the faces of the people who did all this work. And here's some more. There, it can be a lot of fun. On the left, that's just a party of people that are involved in mercury research on my back porch. I also work with other professors. That's John Swaddle at the very top in the hoodie there, and Matthias Loy, and this is one of our end of the semester parties. So it's a lot of fun, but also these life-changing experiences for many of these students actually just lead them right into their careers. So this is 3 students out in the Shenandoah Valley. They're hiding waiting out a rainstorm under the hood of my car there. And all 3 of these students are professionals today. So the one in the middle, some of you might know, Ann Condon. She's been working for the Fish and Wildlife Service. We were studying mercury in birds out there. She's still in charge of contaminants and wildlife here in Virginia for the Fish and Wildlife Service. On the right, that person is a professor at Auburn University. On the left, she's a professor at Albion College. And so they are all doing exactly what they were doing during this research now today, but they're also teaching other people how to do it. So if you are thinking about what William & Mary is and how it differs and is better than a lot of other places, it's that the professors here all work with students. They don't just do their work with the professionals and then teach students in the classroom. It's all mixed together at William & Mary. So the professors' research is completely dependent on these undergraduates, primarily undergraduates, to do the research with them, because we don't have very many PhD students, especially not in biology. We have none. And so the students get a chance to do real research with real results. And for many of them, it's life-changing. Even if they don't become a researcher, they develop unbelievable skills in this area. So I'm very proud of what William & Mary does, and you will find very few places, if any, that do it better than we do, undergraduate research. All right, enough of that. Let's get on with the actual subject here. So mercury in birds, why is it a problem? Let me just give you a quick crash course in mercury. On the upper left is cinnabar ore. Mercury is a naturally occurring metal that is mined in a few places in the world where it occurs near the surface. That map there shows you some of the main mercury deposits in the world. For 3,000 years it's been mined. It was originally used as a pigment because it has a nice red color. But then the metallic uses of it were developed, and it's especially important in the mining of gold and silver, because mercury binds with gold and silver. So when you have rock that you think has gold and silver in it, you can crush it up, mix it with mercury. The mercury will pull all the gold and silver out of it, and then you can burn off the mercury, and you're left with the gold or the silver. So we have spread mercury all over the world through mining of the mercury, and then of the gold and silver, and using mercury to extract the gold and silver, and even through putting it in people's teeth, and then incinerating those people. All that mercury gets released in a place other than where it came from. So now, though, and primarily, there's tiny bit of mercury in all coal and all fossil fuels, actually. But mercury, when you burn a lot of coal, that's a lot of mercury. So we have spread mercury all over the world to the point that tuna fish in the oceans, anywhere, is contaminated with high levels of mercury. And if you want to do one good thing for your health, just stop eating tuna fish, and you'll never have to worry about mercury again, because that's the primary source of mercury for human beings. But for wildlife, they can't just stop eating fish, and so mercury is a problem for wildlife because of the way we have spread it around the world. We have quadrupled the amount of mercury that's available in the food web from what's naturally occurring, and since it's highly toxic, that is a problem. So, are there problems with mercury that we know about for sure, where they're causing problems for birds? Yes, there are a few instances where we already know mercury is a problem without having to do more studies on it. The ivory gull, which is a badly declining far northern species, there are only 500 of them left in the New World, although there's a bigger population in the Old World. They're up above Canada or up above Russia and Scandinavia. Ivory gulls have extremely high levels of mercury because they eat dead seals and whales and the dung of polar bears that have eaten seals and whales. And so they are eating at the very, very, very pinnacle of the food web. And so biomagnification has increased the amount of mercury. And so most of their eggs don't hatch because they have such high mercury levels. So there's a species that is just being done in by mercury, even though it's very, very far away and far from industry. It's because so much mercury is circulating around the world's oceans that it ends up in these ivory gulls. The rusty blackbird is a bird that we're familiar with here in Williamsburg. They occur at William and Mary on the campus, and unlike other blackbirds, they spend most of their time in water, and they eat aquatic food, and that exposes them to more mercury. And so the 90% decline in rusty blackbirds was possibly attributed to mercury, because we don't know why they declined. It turns out that mercury is maybe just one of 10 bad things that's happening to rusty blackbirds, and probably not the most important one. But that's a bird that it seemed it was important to figure out how much mercury they're getting exposed to, because they are slowly going extinct. Then we have a Carolina wren here, which just represents all songbirds that eat spiders. Spiders are little accumulators of mercury because they eat they may eat aquatic food. So the question I've been pursuing for years is whether Carolina wrens and other songbirds that aren't exposed to particular sources of mercury in their diet, but might be getting indirectly through spiders or other predatory insects, are actually at risk. And I'm working on a paper right now that's going to hopefully answer that question conclusively. They're definitely at risk if they're near a mercury pollution site like the Shenandoah River, but are they at risk everywhere from mercury? And finally, the salt marsh sparrows, the salt marsh sparrow and the Nelson sparrow, they are going extinct slowly because of climate change, raising sea levels and wiping out their habitat. They live in salt marshes, and salt marshes are being drowned, and this species will go extinct in the next 50 years or so unless something is done about climate change, because there'll be no habitat left for it. Therefore, mercury doesn't seem like it would be important. However, mercury is very high in these birds, and it's very high in their habitats. If we are going to save these birds, we're going to have to build salt marshes for them, and we have to make sure that those salt marshes don't have high levels of mercury. Because if they do, then it's a lot of effort for no reason. So there's 4 examples of how mercury is affecting birds out there that are not from my research. What was my research on the Shenandoah Valley? Essentially, it all comes down to one thing. We put up 1,000 bird boxes. We lured birds into them, swallows, bluebirds, wrens, and we let them have babies, and then we put a little collar around the babies. As you can see, this little white thing coming off my thumb there is part of the collar around the baby's throat. And when the babies tried to swallow the food that their parents gave them, they can breathe, but they can't swallow food. So then we come running back and we pull the food out of their mouth, and we take the collar off, and we put the bird back in the nest. And through doing that for quite a few years, we were able to figure out exactly what the birds along the Shenandoah River are eating, how much mercury is in every item, and how much mercury, we take a feather from the baby bird and a drop of blood from the parent, and how much mercury they're exposed to. And essentially, without showing you any graphs, this is what we found. Spiders have as much mercury in them as fish when you have a polluted river. And so if you're a little wren, like this house wren here, and you eat spiders, you're going to be just as at risk as if you're an osprey or a bald eagle eating fish. And that's a high risk. And it's a risk high enough to cause you to lose about 20% of your babies, to not be able to remember things spatially, because it affects the hippocampus, and all sorts of other problems. So in a polluted area, little songbirds are at risk because they're eating spiders. Spiders are only about 30% of the diet of songbirds, but they bring in about 75% of the mercury. And that's enough to cause real problems for these birds. So there's 6 years of research right there. Let me just show you what we did was we took birds, we created a colony of zebra finches, which are a domesticated species that works very well in the laboratory, and we put them in an aviary and we exposed them to mercury. And we studied them for years and just recently finished those studies. And here's a sort of a highlight of what we found over the years. We found many, many things, including that the birds can't remember where their food is if they're fed mercury because their hippocampus is affected. But here's the reproductive effects of different levels of mercury. So along the bottom, you see 0 parts per million, 0.3 parts per million, 0.6, 1.2, and 2.4 parts per million. And there's a zebra finch in the upper right. And what the little blue diamonds are is how many babies are produced in 1 year by a pair of finches on these different diets. And you can see that with no mercury, they produce 3.5 babies a year. But with 2.4 parts per million of mercury, which is what swordfish has in it, then they are producing less than 1 baby a year. So that's a pretty severe effect. A population like that would go extinct in the wild. That's the level that spiders are at. And so for this next graph, I'm just going to show you the results of a more amazing finding. These birds are eating mercury while they're having babies. But what happens if you take those babies that were exposed to mercury in their egg and when they're very little being fed by their parents, but then you take them away and you just expose them to that one level that spiders have? What happens when those birds grow up? Now they have no mercury left in their bodies, right? And you let them have babies. They also, if they were exposed when they were little, it's the graph on the right there, versus the controls were not exposed to mercury. This is how many babies they have that grow up to be adults themselves. And this is over a longer period of, it's over 1.5 years. You expect them to have 5 babies. Even in the control group, there's some, you see those 3 dots in the bottom that don't have any babies at all, okay? And there's some that have a lot of babies. But if they were exposed to mercury in the egg and then never again, they have fewer babies when they themselves grow up, about half as many babies. And a lot of them have no babies at all. So the effects of mercury are not just on the birds that are eating it themselves, but on their babies, even if they migrated away and started a life somewhere else, far away from the Shenandoah River. So that's, to me, that may be the most amazing finding, totally unknown beforehand. Okay, so in the interest of time, I'm going to skip this slide and just tell you that this work, while very basic, also has strong applications. So our research was used as part of the lawsuit against DuPont, which polluted the Shenandoah River, for a $50 million settlement. That's not for cleanup. That's just to create habitat for birds and other wildlife, and fish especially. So they literally used our data to come up with this number. And that is very exciting. And for the students that were involved in this research, it was incredibly exciting to be involved in something that really mattered. Literally just within a few years of them finishing the study, they could see the results. This is not something where, you have to wait till the very end of your life and then maybe someone figures out why your basic research was interesting. The money will be used for projects to improve bird habitat along the Shenandoah River, including that parking lot that I'm standing in there. That is slated to be torn out. That's the parking lot of the actual factory that put the mercury in the river. That's slated to be torn out and turned into a forest for bird watching, like a park for bird watchers. Whether that actually happens, we'll see, but that's several $1 million of the money went to that. So that's really exciting. Since doing that research, I have been involved in a lot of other rivers that have pollutants like mercury, but also PCBs and dioxin. And that's really what my research is now. It's moved away from the things I've been talking about. And so here's two other cases that I was involved in. This one is on the Penobscot River. I can talk about these because their settlements are public. Here, the company Mallinckrodt, which put mercury in the Penobscot River, has to pay between $187 and $267 million, not to clean it up, but to improve bird habitat. Cleanup is a different court case. And I'm also involved in the Hudson River case. That doesn't sound like much money, and it isn't, because the company GE that polluted the Hudson River with PCBs has already spent $1.6 billion cleaning up the river. But this is just one tiny part of the settlement, $20.5 million for further studies. So I'm doing some of those studies on the birds to try to see how much exposure there was, how many babies they're having, etc. And I'll just finish up by talking about the projects that I'm working on literally now. This one is on a river that is contaminated with dioxin. And on the left, you can just see just what a strange environment it is. You're looking for birds in the shadow of these incredible industrial facilities, the very same ones that put the contaminants in the river. What my job is to find out how many birds there are there, how much of the pollutant is in their eggs, so what is their level of exposure. As a result of that exposure, how many fewer babies were born, going back to 1981, which is when the laws that cover this went into place. The companies get off scot-free before that. And then that allows the government to pursue the company in court or for a settlement to try to remedy that situation. All those birds that weren't born belong to the people, to the American people. They belong to you and me, and the company is responsible for replacing them. And so that's what these huge settlements are about. Figuring out how to make more birds for us to replace the ones that the company took with their pollution. On the right here, just as a matter of interest, is a nest of gray catbirds. Those are very healthy little baby birds. Well, I don't know how healthy they are, but they're perfectly alive. And that nest is entirely made of plastic, as is every single nest at this site. Gray catbirds are extremely common here. They love this habitat because there's maybe fewer predators. There's a wide enough band of shrub that they really can find places to nest. Apparently they can't find enough grass and twigs to make a nest, but there's plenty of plastic, and every single nest looks exactly like this. I didn't just pick a weird one. Other species have different kinds of plastic that they use in their nest. None of them does quite what the catbird does, which is to make the entire nest out of plastic. Just one more site. This is the site that I'll be working on this coming summer. This one's on the Mississippi River, and this one is really something. At those other sites, the contaminants are in the mud. You don't see them. Here, you just turn a corner, and there they are, just sitting there in barrels. The company that owns this site manufactured almost all the PCB used in the entire world at this site. And so when they have a bad batch, they just dumped it in a creek or put it in barrels and threw it around behind somebody's house. So this is going to be a very interesting site to work on. And I'm going to just finish up by just talking about my father for a moment. there's a picture of him on the left, and he is actually with the guy that was in the first picture of me where I said, notice all the hair, all the dark hair. And so that was my dad coming out of his last bird walk. I lost him in September. And so I wanted to dedicate this talk to him and to thank you all very much for having me. And many of you met my father, I think, and he came on some of the Williamsburg Bird Club trips, and he always was thrilled to get the news about the club and to hear what's going on in Williamsburg. So I know we all miss him, and I think my mother's on this call. So thanks, Mom. And I guess I'll take any questions if there are any.
Well, thanks, Dan. That was just unbelievable. And I do remember being with Chesapeake Bay boat trip, and we all decided if we stayed close to your dad, we would probably see the best birds.
Yeah.
So I think we tailed around behind him for quite a bit of it. I think there are some questions in the chat room. That was just I have pages and pages of notes. I wouldn't even know where to begin to start asking questions, but I think there are some in the chat box if you want to take a look.
Okay, I'll take a look.
Okay. Okay.
All right, Shirley Devan gets a prize because she guessed size 7 before I announced it. So good job, Shirley, on the gulls and the clams. One question from Diana is, which golf courses were better for birds, surrounding areas, or something else? That's a good question. I won't get into naming specific golf courses, but the design of golf course that is best for birds is if they preserve a lot of forest around the golf course and don't put houses around it. So the common thing of building a housing development and putting a golf course right in the middle of it is not the best for birds, because the golf course itself cannot support them alone. They need to move off the golf course when they're young into surrounding forests, or they will get eaten by hawks. And so a golf course is a bit of a death trap for young birds. And so when you design a golf course, or if you choose which golf course to play at, choose one that has preserved a lot of woods around the course and not put houses right on the edge of the course, because that's not good. Next question, what is being done to help birds settle in the newly created wetlands? That's a great question, Judy. Nothing is being done. The problem is that the people building these wetlands are being forced to do it by the Army Corps of Engineers. And strangely, they are put in charge of monitoring their own wetland. And they're not really motivated to see that it's good for birds. They just need to make sure the plants are alive, and that's where they leave it. So created wetlands are designed to make sure that the plants survive, which means that they're a little bit too wet. And so that makes them sort of like ponds instead of marshes. And so they're good for Canada geese and wood ducks, but they're not really good for the birds that were in the original wetlands, which is all the songbirds that need marshes and swamps. And so the system is not good for this, and the system could be improved by either making birds a priority or having an outside service that assesses wetlands, but by letting the developers themselves assess the wetlands, and by having it assessed only on whether the plants survive or not is leading to problems. Where is the mercury site in the U.S.? That's actually around the Bay Area. The gold rush was largely possible because there was also a source of mercury there. So they could mine the mercury and use the mercury to mine the gold and not have to bring the mercury over from Europe. A lot of mercury was brought over from the old world. And in fact, some of those ships sank, and there are sunken ships with flasks of thousands of pounds of mercury still in the oceans today, probably slowly leaking out from shipwrecks back in the 17 and 1800s. Where is the river with the dioxin? Well, it's in the mid-Atlantic. Because these are legal cases, I signed confidentiality agreements, and until the settlements are done, I can't really talk about the rivers. But there are multiple rivers with dioxin in them. The one I'm studying is not in Virginia. And someone says, and speaking of bluebirds, you said most birds die within their first 3 months. Was that statement referring only to bluebirds or to other species? Well, it's actually referring to all species of songbirds and many other birds. The most dangerous time for a bird is after they've left the nest, not right away because their father is still feeding them during the next 2 weeks. But after that, they're on their own. They have to learn how to feed. If it's a hawk, they have to learn how to catch prey, which is extremely difficult. They might be able to catch a few things, but not enough to feed themselves. So those first few months before migration and during migration, when they're making their first migration by themselves, are the most dangerous for birds. And most of them don't make it. If they did make it, then the population would double and triple every year, and we'd be seeing more and more birds. But in fact, the populations stay about the same. Most birds live a couple of years, which means most of their babies don't make it. And for a population to stay stable, once every couple of years, every pair of birds has to have two babies that survive to become adults. And so that's approximately what you can expect to happen. It's sad to think about. And it's especially sad when you see an adult bird killed against a window, because populations really can't afford that. Every bird that gets killed is a tragedy. Just because a lot of them die doesn't mean we shouldn't feel bad about the ones that get killed by cars and windows and all that sort of thing, and dioxin and mercury. And as you all know, the bird populations are decreasing sort of across the board, except for vireos. Every single songbird group is declining. Has declined by about a third in the last 50 years, and probably had already declined a lot before then. So birds are not doing too well, except for a few groups. Are there any other questions? That's all I see in the chat.
Dan, I can't help but ask, do you have a favorite? Do you have one of these research projects has been like, it?
That's a good question. Let's see.
I didn't mean to put you on the spot.
My favorite project that I ever did, the most exciting project I ever did, was in high school, when I put up a bunch of birdhouses for screech owls, which I'd never seen one. And we had to do a project as a junior in high school. So I went all over the city and I put up these birdhouses that I made. It took me, weeks to make birdhouses. I didn't know what I was doing. And I think I was hand sawing the lumber. And then I checked them all winter. There's nothing but squirrels. And then one day I opened the box and there was a screech owl in that box. It was sitting on top of a huge pile of feathers. It was making a nest. It was all flickers and cardinals that I think the bird had eaten and just thrown into its box. And that was probably the most exciting moment of my entire research life. I haven't quite matched that yet. So
I can imagine. That's a good one. Well, thank you so much. Anybody else have? Oh, go ahead. Somebody have a question?
I was gonna ask Dan about chasing the eagle in Maine. How did you find it right away, or what was that like? Was it a whole crew of stalkers there looking at the bird?
Oh, it was something. So I actually wrote it up for my Saturday column, so there's more details in that coming up. But basically, just deciding when to go was the hard thing, because it kept moving and disappearing for a week. And then I started to go, and it was the day that I-95 was completely stopped for 26 hours, so I didn't make it out of my driveway. And then the bird disappeared that day anyway. And I happened to have a student up there that day, and they said they were looking at it. And then later in the day, it turned out that the bird that everybody saw was actually just a branch in a tree. It was 2 miles away on an island, and they were all looking at it in the scope, and it wasn't even the bird. It just stayed there all day. And then someone realized by looking at photos that hundreds of people think they saw it, but actually just saw a branch in a tree. So it disappeared for a few days, and then it showed up again, and I just jumped in the car and drove up there. I mean, I literally didn't even, when I came home, I realized I hadn't taken the trash out and the whole house smelled. I just jumped in the car and left. And I drove to Maine. I picked up that Nathaniel, my friend in Philadelphia, saw my mom, and went up there. We got there late in the afternoon, and there was all these people standing around forlornly. No one had seen it. They'd all been there. This is their second or third or fourth try. And so we didn't see it. And we came back the next morning, and it was horribly cold. I mean, it was 1 degree, and it was just snowing and foggy, and everyone's just so morose and sad, and they were anxious. They were like, I tried 5 times. I better see it. And there's all these rumors someone saw it here, someone saw it there. But the problem is it's all private property. You can't go walking through the woods looking for it. It's just people's houses. So you just have to stand somewhere and hope that it flies by. So we're all standing in this parking lot where you could kind of see a lot of the river. Anyway, it didn't show up. So we wandered away, my friend and I, and then we found a nice bridge and we stood on it and we were just kind of enjoying ourselves for a while, and the bird just flew in front of us. Nobody else was there. It was really spectacular. And then it didn't get seen for a couple days, but then it came back and lots of people saw it. It was kind of just sitting right there by the parking lot. So it's still there. But if you do go for it, give yourself a couple days, because it's not easy. You just have to be lucky. But everyone gets lucky eventually because it is there. And there's no mistaking it, that one. You don't even need a bird book. You don't even need to go look at a picture of it. You'll know what it is when you see it.
Thanks.
Any other questions or comments?
I have a
Yes.
question. So I wanted to know, are the settlements you're talking about under federal Superfund law, or are they state law, or both?
Great question. They're under federal law, but it's not Superfund. Superfund is for cleanup, but
Yep.
these are under what's called the NRDA,
NRD.
or Natural Resource Damage Assessment Act, or something like that, or which is only for replacement of wildlife resources like fish, shellfish, birds, to the
Right.
people. So these settlements are just for that, and they don't go to cleaning up. Cleaning up is the EPA. This is the Fish and Wildlife Service.
Right.
So the companies get hit with multiple laws and multiple lawsuits from different agencies.
Thank you.
Good question.
Well, that was a absolutely fabulous presentation, Dan. I think it makes me wish I was a student. And if you ever need any help, I think there's a lot of willing people that would be happy to get out there.
Great.
So
Well,
thank you.
I will just make a plug, since you just said that I truly believe in William and Mary and in education as sort of the, our only way out of this mess that we're in, and environmentally. And so if you don't know what to do with your money, you can give it to the Ruth Beck Fund at William and Mary in the biology department, which is specifically for bird research. Or you can give it to the Williamsburg Bird Club, which has a Ruth Beck Fund also that funds these students. And a small donation to William and Mary for bird research goes a really, really long way. So I'm sorry to be aggressive like that, but that is something that people can do. And
Great.
Thank you so much for listening and for making this research possible over all the years. And for all you other people from other clubs, please do something like this in your own club and try to, support the bird researchers in your own area. And thanks so much for coming out.
Thank you, Dan. That was terrific. Thanks so much. We're going to do just a couple of quick announcements. The guest